Good morning, everyone. We're going to be presenting on the DNA module. So before we go to our quick video introduction, I'm going to be introducing my other two group mates.
Bella Yang. And I'm Giovanna.
Change our grades to 100, and then everyone else, we just, we give them zeros, so we can be the best. We're winners. They deserve a zero.
Two. Let me check my student focus out. Suspicious.
I saw you with the professor and we have to meet with him right away.
Do you three have anything you'd like to admit to me? Really? Because I find these 3 grades here very suspicious. I demand cheek sets.
Okay, so that's an introduction. So now we are trying to figure out which one of the 3 of us are guilty in changing the student grades. So before moving on, so what we're gonna have to do is a data analysis, right? DNA analysis to figure out, right, who it is that's guilty. So before moving on, I just wanted to briefly introduce just some of the very basic terminology regarding DNA. And so what is microsatellite DNA? So it's actually short tandem repeats, right, STR markers. And as you can see in the strand that we're showing here above, it's a repeat of GGCA. And the predominant mechanism for variation in the number of repeat units is actually through the mechanism called slippage. And we actually have a very similar figure to this one over here in our book. And so over here you start with 15 CA repeats, and then when DNA is replicated, the polymerase comes in and it's replicating normally, and then it pauses in the repeat domain, and then when the DNA melts, it'll reanneal incorrectly, and so you see over here, there are some extra repeats over here, and then when the mutation is repaired, it's actually going to be repaired incorrectly, which will lead to 17 repeats versus the 15 that you started with. So that's how you get more and more repeats. And so the STR markers that are used for forensic purposes, there are normally 4. They're tetranucleotide repeats, such as the GGCA right there. But as you can see over here, you can also start with 2. You can have 2, 3, however many. And then also a homozygous genotype is when you have the same number of repeats in both of your homologous chromosomes, such as 5, 5. And then heterozygous is when you have a different number of repeats, such as 5, 7.
Invented in the 1980s, PCR is a way to replicate a specific strand of DNA in a cell-free environment, and more than 1 million copies can be made in just 3 hours. The 4 main ingredients are the original DNA strand as a template, 4 deoxyribonucleotides, DNA polymerase, and a pair of single-stranded DNA primers complementary to the flanking sequence. A good primer is between 17 and 35 base pairs, between 40 and 60% cytosine and guanine. And PCR alternates between 3 temperatures, the first being 95 degrees, which splits the first strand into 2, then 55 degrees, which allows the primers to bond, and then 72 degrees, which allows DNA polymerase to bond and create the complementary strand.
So the method of gel electrophoresis. So this is a very common way to separate a mixture of DNA or other proteins according to their different molecular size or weight. So basically, at first, the negatively charged DNA started at a negative end of the electrophoresis chamber, while an electric charge pushes DNA fragments through a porous agar solution. And the larger and heavier DNA fragments, they tend to travel a longer distance, while the smaller ones, the larger and heavier ones, they experience more drag and travel a shorter distance, while the smaller ones tend to travel a farther distance. And the perfect agar solution shouldn't be too thin or too dense, because if it's too thin, then all the fragments tend to travel a very long distance, so you can't really see the difference. If it's too dense, then all the fragments experience a huge resistance while they're trying to make it to the other end. So they'll eventually heat up and possibly denature. And the loading dye is required to visualize the colorless fragments. And the fragments of the same size will be organized into bands and eventually form matter.
Okay, so we have performed the analysis now, and you can see over here is the electrophoresis results, and then here's the electropherogram. And so over here we actually only showed the first part of them, just because we can't fit everything into the slide. But just by looking at this, can you as the audience tell us who is guilty? You could just shout it out.
Evan.
Yep. It's Evan. And you can see, just because the bands look exactly the same, over here you see the peaks look exactly the same. So yes, Evan, you are guilty. And so,
According to DNA evidence and evaluation, you are guilty. What do you have to say for yourself, young man?
No, not. It's not me. It could be anybody else.
I know exactly how to prove to Evan that he is guilty.
So then we move on to the Hardy-Weinberg equilibrium. And so the professor decided that this was the best way of proving to Evan that he is guilty and it can only be him. So what he did was that he used a table, he converted the fragment size, and remember that you did do this in lab. So he converted the fragment size to the allele, and all of this, again, is using a table that is given to you. And then you determine the genotype, if it's homozygous, heterozygous, And then you record the allele frequencies for Evan's ethnic group and calculate the genotype frequency using the frequency formula. And remember that the frequency formula is over here for heterozygous, and then you square it for homozygous. And then you multiply all of these frequencies here to estimate the random match probability of Evan's profile. And then you take the inverse of this number here to get his genetic profile frequency. You know that these are real values. And so, considering the current world's population is 7.5 times 10 to the 9th right now, as of last week, and the chance of finding someone who has Evan's same exact genetic profile is 1.13 times 10 to the 10th, Evan is guilty. So, there we go. That's it. And our credits are to Alex Wolzanski. He's here this morning. He was our director, cinematographer, and editor. To Victoria Biancavilla, she was playing our professors. And if you know, he's wearing the same color shirt as she wore in the video today. Thank you, that was great. And also Angela, our awesome TA, and of course, the professors. Thank you.